Okay, so the first impression I got when I got my hands on the B4B book was that it was very comprehensive, but there was something missing, namely metagenomics. And then the second impression was that there was some kind of biomedical flavor in the book. And then I thought a way to bring the two things together would be to talk about, to introduce metagenomics by way of talking about this particular problem, biological problem of the microbiomic basis of disease. And the way I thought about structuring this chapter proposal was with some background material on basically the structure of ribosomal RNA and lineages and taxonomies for microbial identification, and then two particular problems in some detail. One of the problems and computational methods associated to microbial identification on the human body. And then the problems are, we'll see in some detail, of alignment, some recurrent problems in bioinformatics alignment, then the taxonomic assignment, and of next generation sequencing rates. And then a first part of assessing the microbial diversity in a sample. And then another kind of problems is the comparative analysis of different microbial communities, which appears also recurrently when comparing microbial communities in health and disease, across time, across space, but okay, the computational problems are more or less the same. And then some bibliographical notes. So a little more in detail. For the background material, apart from introducing the subject of metagenomics, the focus was to relate it to phylogenies and taxonomies. And in this sense, to introduce a little about the techniques used for sequencing the metagenome, and how to place it in a phylogeny or in a taxonomy. For the first part, here is I was trying to avoid any overlap with the current first edition, and also with, I don't know, with possible chapters you might be considering. Here, what is needed at least is to explain the structure of the ribosomal RNA, universal primers for PCR, what enables the amplicon-based metagenomic surveys. My initial idea here was to also take this opportunity to explain the design techniques for the design of PCR primers and based on suffix arrays, but then I thought that it would be too much material for this chapter. Maybe it's already covered somewhere else or is planned to be covered somewhere else. Anyway, another thing that is really important here is taxonomies. So biologists are already familiar with King Philip, etc. But there are still many things that are very relevant here, like LCA algorithms for mapping sequencing reads and placing them in resolving ambiguity. So I will briefly explain that in a little more detail later. And then for the analysis of microbial communities in the human body, well, this is just a biological introduction, that microbes are not necessarily bad. We need them for many metabolic functions, and there's there's been this relatively new subject. Metagenomics is only maybe 10 years old or so, but now there's an explosion of metagenome sequencing projects and papers dealing with computational methods, with biological findings coming from these metagenome surveys, and they're starting to appear more and more papers about these metagenome surveys related to health and disease. For instance, studies of the gut microbiota showing which are the dominant genera phyla in health and disease, in particular diseases, and the same for the skin microbiota and for the oral cavity microbiota. And so this is the biological side. So what's the computational side to all this? So how can we do all this? And this is a more risky part in this charter proposal, because these are all, I tried to pick what I consider the most well-established methods. But since metagenomics is relatively new, this could change on time, or maybe there are still new methods to, there's still a blast for metagenomics to be discovered, I don't know. But my best guess right now is that regarding that there are these three particular problems to be addressed here in the analysis of microbial identification. One is the alignment of short reads to microbial genomes, either full genomes or full 16As. And as computational methods, I think that most of the tools being used now to do this efficiently are based on suffix arrays, barrows will transform, and some variation, Faragina index and so on, but at least it might be appropriate to discuss this at this point in the chapter. If there's no overlap, maybe suffix arrays already appeared many times in the book or will appear. So this is the first part, alignment. Then is the problem of the taxonomic assignment of the sequencing reads. And then there's computational issues involved here. One of them is that it's not always possible to identify microbial species in a sample at the species level. Sometimes there are or most often there are ambiguities, and because sequencing reads are short, and or are shorter than the full genome, shorter than the whole 16A sequence, and these ambiguities are usually solved in most tools by assigning the sequencing reads to the LCA in a taxonomy of the matches. But there are better ways to do that, and I worked on that. So in particular, so this is maybe the only place where I would like to explain more in detail in the book, in the chapter, what I did in metagenomics, and the other. I think there's no way to make it disappear. And another aspect is how to quantify the diversity in a sample. And here there are many possibilities and many measures of biological diversity coming from ecology. And the notion is that of alpha diversity, so diversity within a habitat or sample in this case. And two of the most widely used and simplest to compute measures here are the Shannon-Wiener index, which is just the information entropy of the sample. And the other one is the Clark-Warwick index that takes a taxonomy into account. And so here the computational problem will be, method will be just to explain how to find efficiently paths and compute paths in a microbial taxonomy. And the other biological problem here was the one of comparing microbial samples coming from the same individuum in different states of health and disease, or across time, or even completely different microbial environments. So the problem is to detect and quantify change. And here again, there are well-established measures of diversity coming again from ecology. And this now beta diversity, so diversity across environments. And what I consider the best measure proposed so far is relatively new. It's the Unifrac Index, developed in the Rob Knight's group in the US, and it's based on the proportion of the clades that are that are unique to one environment only. So what the species that make a sample unique. And so again, here it's the computational methods to be explained in this chapter, in this part of the chapter, are simple tree traversal algorithms for traversing the clades in a taxonomy. And then this could be supplemented by bibliographical notes for each of these biological findings related to metagenomics and also the computational methods. And to finish, I mentioned here this problem of species identification at the best possible taxonomy rank. So in case you are not familiar with these notions of taxonomic assignment, let me just switch to another presentation. So the idea is the following. When given a set of sequencing reads, they are the first step to identify where they come from, given a reference database of known microbial sequences, is to align the reads to these sequences. And then the result of the alignment is a set of matches, say within some threshold of sequence similarity or number of mismatches or whatever you choose, depending on the tools that you use. And then there these are the ambiguities I mentioned before. So the sequencing read might have come from any of these species represented there in green. And the usual way to solve this, to resolve this ambiguity, is to assign the read to the lowest common ancestor here of the matches in a reference taxonomy. But this introduces bias and error because it would also cover a number of mismatches, which can be very big in a whole taxonomy. So this LCA mapping is conservative, but usually introduces much error. So what I have worked on in taxonomic assignment for a while is from different points of view, and the best results that we got, they are implemented in a tool called Tango, Taxonomic Assignment in Metagenomics. It's based on considering the same scenario, but instead of assigning necessarily to the LCA of the matches, consider what happens, consider each possible node in the taxonomy below that LCA. And here In the first case where we consider only the LCA, the matches are true positives from a classification point of view, and the non-matches are false positives. And there are no true or false negatives here. But in this case, for any other candidate node in the taxonomy at a lower taxonomy rank, this is the if this is the subtree rooted at that node, we have instead of the whole green set of matches, it only covers a subset of this. These are now the true positives, and the other ones are false negatives in this case. And the other way around, from the non-matches we had before, now we have a smaller subset, which are the false positives, and the others are true negatives. So by considering this for each candidate, we can solve an optimization problem and combine these true positives, negatives, false positives, and negatives in any way you want. So we have two ways to do that in this Tango tool, and one is by means of the F measure from information retrieval, and the other one is by means of penetrative score that uses a parameterized score by which you can decide the weight to give to the specificity of the assignment. And then on one particular case, for this parameter equals to 1, you get an assignment at the LCA. For the parameter equals to 0, you get an assignment at the leaves, at the species level. For this parameter equals 0.5, you get an assignment identical to the F measure that optimizes this scenario. But you can play with this parameter, any real value between 0 and 1. Okay, so this is what I think is the best solution so far for taxonomic assignment. Okay, thank you.
Questions? I have a question actually. Do you plan to cover single cell genomics, this application of genomics?
I think it would eventually it will have to be covered, but I think it's perhaps not the right time yet. It's too early yet.
Yeah.
But it will be very important. Yes.
So, what level do you cover the suffix between self-revealed and the bonus will transform business for biologists?
I think the biologist doesn't need to be involved in the details of implementing suffix array algorithms in linear time. So, but the notion of suffix array, how you can build easily a suffix array by sorting, just sorting suffixes, and then of course you won't do it in practice like this. But the biologist, I think it's important that he knows the notions behind that. And at the end of the day, it will be using tools that are software engineered to be able to process millions of reads in minutes of CPU time. So it won't be implementing an aligner. So I think the focus should be on the concepts rather than the efficient algorithms at this level. So for other things, for tree algorithms also, I think it might be more sensible to, more interesting to introduce at some more detail the algorithms.
Most people that study metagenomes really also use different cluster techniques, and I think these techniques is quite important for the biologists that want to study bioinformatics. So have you considered maybe to include some?
I think I try to keep the focus here on amplicon-based analysis. I think that the use of clustering metagenomics is most often when it comes from shotgun sequencing. But still, it could fit here at some point, but not necessarily. So it's a choice to be made. I think that there's already enough material to have a relatively long chapter, so that's why I'm trying to cut it down a little. Thank you.